Okay, so welcome to CHEM431B. This is your last lecture before the midterm on Monday. So I have a handout for you that talks about accounting for all the ATPs that are synthesized in the cell. We'll go over that handout. I'll also talk about ATP synthase, this amazing enzyme that creates ATP from ADP, an inorganic phosphate, and it uses proton motive force in order to drive that reaction. I'll talk about inhibitors of ATP synthase. ATP synthesis. There's a variety of different types of inhibitors, and they belong to different classes. And it's these inhibitors that helped us elucidate the electron transport chain and the production of ATP and how we got to know the intimate details of this process, oxidative phosphorylation. And then I'll talk about a relatively new phenomenon where the electron transport chain, and specifically cytochrome C, is involved in a process known as apoptosis, also pronounced apoptosis, and that means programmed cell death. So I'll talk about cytochrome C's involvement in that. I want to remind you that we have a midterm on Monday, chapters 12, 13, 14, and 15, basically where we left off for midterm 1. So all the chapters that we discussed since then. Please bring a calculator and also bring a ruler. And let's go ahead and hand out the handout so you guys have it. Okay, so we'll get to this handout in just a bit. We'll discuss a little bit more about electron transport chain and ATP synthase. So ATP synthase, as I said before, is composed of two protein complexes that come together. And does anyone remember where the location is for ATP synthase? Exactly where in the cell do we find it? Mitochondria, very good. Let's get even more specific. Where in the mitochondria? That's right, the inner membrane of the mitochondria, not the outer membrane, but the inner membrane. Very good. Okay, and I'll just go ahead and, okay, wait a second here. How do I go down? I'm using a new type of program here called Journal, and I don't know how to. Okay. Okay, so let's see. ADP plus inorganic phosphate plus a proton, and that's going to give us ATP plus water, and this requires proton motive force, as I said before. And it turns out that it was discovered by this guy named Boyer, which I worked across the hall from, that the driving force for this PMF is not actually in the production of ATP, but in the release of ATP from the enzyme. So actually all of this occurs, the ADP and inorganic phosphate forming the ATP, that occurs on the enzyme without any kind of energy. You don't need energy to create the bond between inorganic phosphate and ADP, but you do need PMF to release the ATP from the enzyme. So basically, you've got two steps here, ADP plus inorganic phosphate plus the proton, and that's going to give us ATP. I'm going to say bound to enzyme. Okay, so it's going to be bound to the enzyme plus water. Okay, and then what Boyer did was something very interesting. He said, okay, if I can create this on the enzyme without any kind of ATP, then I should be able to drive the reaction backwards if I have ATP loaded onto enzyme, and if I use radio-labeled H2O, so this was his key thing, he used water that had an oxygen that was an isotope that had a mass of 18, so that's that's radioactive. And then what he could do is basically find that the inorganic phosphate would have the oxygen incorporated into it, and that's exactly what he did. So again, I'm going to have to move down. Okay. You guys better be taking good notes here. I'm not sure about this program. So this is what he did. He took ATP that's enzyme bound plus H2O, and then he was actually able to create ADP plus the proton plus, I'm going to write out the inorganic phosphate here, plus inorganic phosphate that had a single oxygen that had O18 in it. So phosphate has 4 oxygens, but one of the oxygens had O18 in it. And so this proved to him that he could actually go backwards with this reaction, and actually he could catalyze this reaction several times. He can go backwards and forwards and keep on incorporating water into the phosphorus. This told him that the ATP was synthesized in the absence of proton motive force, and the proton motive force is required to release the ATP from the enzyme. So that was an exciting experiment, which as a graduate student, I couldn't understand what he was talking about when he said this at UCLA, but now, as a professor, I get it. Okay, so let's go ahead and show you what this looks like in your book. So let's talk about the overall structure of this guy. So this is on page 650 of your book. And you've got this sort of a side cut, but you've got the F0 part, which is membrane, it's embedded in the membrane, okay? And then you've got this F1 part that I talked about, which is pointed, not in the membrane, but in the matrix of the cytosol. And so what happens is that the protons will actually go through this. Okay, they go through this C10 part, and you can see this almost looks like it could rotate, and certainly it does rotate. And this gamma, just remember this gamma here. This gamma is like the rotor, and it will rotate like a gosh, what rotates now? Everything is so digital. Like a barber shop symbol. It rotates, okay? So it rotates, and you can see that it's kind of lopsided here. And as it rotates, it squeezes these beta subunits, and then these beta subunits will release the ATP that's being synthesized here. So it's a pretty neat system. And you can imagine that the protons need to get through this F0 part, and the way they do this is show you this. This is on page 653 of your book. And these protons will bind successively to glutamate and aspartate side chain. So remember, those side chains are negatively charged. And so these protons are basically being transferred from one of those carboxylic acids to another, okay, until they finally wind their way through to the matrix. So here's the proton coming in, then it rotates through that little rotor, and by doing that, it actually turns this gamma subunits. This is on page 653 of your book. It's a very interesting system. And then the protons make their way back to the matrix. Well, what's happening from a top-down view? So now we're going to pretend that we are looking top-down into the on top of the F1 part, and we're going to be looking at this. And there's actually 3 of these what we call beta alpha subunits, okay? And so that means that there's 3 catalytic sites here. 1, 2, 3. There's 3 catalytic sites, okay? And you can see how this gamma is here. Now this is a fairly complicated diagram. I think they made it more complicated than necessary. All I want you to know is that the ADP and inorganic phosphate, they will bind to one of these, okay? And they will form the ATP. That doesn't require any energy. But when this guy turns, because of the proton motive force, then what happens is that this is actually released. This ATP is going to be released. Okay? So that is the sort of magic of how ATP is synthesized with this ATP synthase. A few years after this was proposed by Boyer, a group, Yasuda et al. From Japan, went ahead and thought that they could create a way of visualizing this rotation. And this was a phenomenal experiment. But what they did is that they created an artificial system where they have a cover slip. It's a glass cover slip they use for looking at cells under the microscope. And then they've got this sort of nickel NTA coated bead. And then they attach to that the alpha, basically the alpha beta subunits. Here's the gamma subunit that I talked about. And then they have artificially put in this streptavidin, which is not natural, and then this actin filament. And the reason why they put an actin filament in here is because actin is very long. It polymerizes to be very, very long. And then they put a fluorescent marker on this actin filament, okay? And what they saw under the microscope is that sure enough, if they put in ATP, they could drive this thing in the opposite rotation. They put in ATP, and this whole thing starts to rotate. This is one of the first experiments showing how a single FTP synthase actually works. And so these are just sort of video clips of what's happening. You can see that it's here for a while, and then it starts to turn, and it turns into all these different directions. Okay, so it actually rotates. And if they limit the amount of ATP, they could actually get it to stop. And then if they put a little bit of ATP, it will go like 120 degrees, and then it will stop again. They put a little bit more ATP, and it will go another 120 degrees. So it's a beautiful way of demonstrating that the early studies by Boyer and others was exactly right, that you got this sort of 120 degree separation between these catalytic sites. So it was a big tour de force type of experiment showing how this ATP synthase works. Okay, they used to have this video online. I looked for it. It looks like it's been taken down, so unfortunately I can't show you the video. Okay, I want to talk a bit about inhibitors of ATP synthesis. And you'll be surprised to learn that some of these are pretty familiar. They actually belong to 4 different classes. One of them is going to be electron transfer inhibition. Another one is ATP synthase inhibition. A third one called uncouplers. And a fourth one is ATP ADP exchange inhibitors. So these are the 4 classes of ATP synthesis inhibitors. Let's talk about these one at a time. Sorry about this. I wish I could just scroll down like my other one, but anyway, experimentation. Let's talk about these ATP, these carriers, the electron transport inhibitors. Your book actually has a few of these listed. I'll show you this one right here. Okay, this is on page 635 of your book. When you notice a couple of things, you should be familiar with these now. Here's NADH, which is obviously going to go to complex 1, and then you go to FMN, and then we have one of these iron sulfur proteins, and then it goes to coenzyme Q, cytochrome B, and then actually this is part of complex 3, cytochrome B, this iron sulfur protein then, here's cytochrome C1, so all of this is part of complex 3, and then here's cytochrome C by itself, and then we've got cytochrome A and cytochrome A3, which is part of complex 4. So it turns out that there are these inhibitors that stop these at different places. So rotenone is actually found in the roots of some plants that grow in Brazil along the banks of the Amazon River. And what native people there have been able to do is if they want to catch fish, they'll take those plants, and the roots are stocked full of this rotenone, and they'll sort of crush them on the banks of the Amazon River in areas where you have slow moving water. And this poison actually that gets in there will actually slow down the fish movement. So it actually slows them down. It makes sense, all right? These fish require ATP to swim. And so now that ATP is not being synthesized anymore because you have no more electron transport, okay? Because you have no more electron transport, you cannot make a proton motive force, okay? So that slows down the fish. They get really sluggish, and then they go ahead and net it. They go ahead and net these fish, and they go ahead and cook them. And I'm not sure how they get rid of the poison, but they go ahead and eat the fish. So they have figured out that there's something in these plant roots that inhibits the fish movement. And it's been isolated now, and this rotenone is that. There's another poison that's used as a rat poisoning called amytal. And amytal is also used to inhibit the electron transport chain. Let's talk about this inhibition a little bit more. If we inhibit the electron transport chain, then it turns out that everything upstream of that inhibitor is going to be reduced. Why is it going to be reduced? Because you have plenty of NADH. Okay, so NADH keeps on feeding into the system, but all of this is going to be reduced. Ignore these blue things underneath for now. Okay, pretend they're not there. Okay, so complex number 1 will be reduced because you've got rotenone amytal. What about downstream? When I say downstream, that means more towards the oxygen side of the electron transport chain. What happens there? Well, it turns out that all of these will be oxidized. Okay, there's just no more. All the electrons are used up by the oxygen. Okay, and when you have this block here, all these are going to be oxidized. So it's going to be complex 3, coenzyme Q, complex 4. So all of those are going to be oxidized. It turns out that there's another inhibitor that you may have encountered at some point called Demerol. So Demerol is an anesthetic, okay? And that also inhibits this electron transport chain. I think it inhibits it right after coenzyme Q. I need to check that, but I think Demerol is another inhibitor. And again, anything upstream of the block will be reduced, in other words, electron rich. Everything downstream of Demerol will be oxidized. Here's another, this looks like an antibiotic, antimycin, which inhibits this part of the reaction. That's going to be complex 3. And then here we have some things that are very familiar also. Here's cyanide, which you've heard is obviously a poison, okay, very powerful poison. That will inhibit complex 4, okay, so it inhibits right here. So you will not be able to continue electron transport because it blocks it, okay, so you will be killed with cyanide. Carbon monoxide, you find that in car exhaust, actually, right? And so one of the ways that people want to hurt themselves is by taking a car and hooking up a hose to the exhaust and then having that hose go into the driver's window, okay? You roll up the window and you turn on the car, and then eventually you pass out and you die, right? That's how you do that. It's really a bad way of committing suicide, by the way, because the carbon monoxide will also have to poison all of your hemoglobin as well. So it's a really slow, miserable way of dying. Given my choice, I would take cyanide. Cyanide is very specific, and so it's much quicker, okay, than the carbon monoxide. Now, this is recording. Okay, I'm not suggesting anything. This is only illustrative purposes for the purpose of biochemistry. I'm just showing this for biochemistry. I'm not. Okay. All right. We won't talk about this anymore. Okay. I also want to show you that we've got these numbers underneath here, and these should be familiar to you. Does anyone recognize these numbers that are underneath here? What do these numbers symbolize? Energy. Let's be very specific. What are they? Voltage. Very good. These are voltage, and they stand for what exactly? Standard reduction potentials. So these are the standard reduction potentials. And again, you can see how they gradually increase as you go from NADH all the way to oxygen. Okay, so those are the inhibitors there. I should say that there are some artificial acceptors as well that biochemists have figured out. So these are the acceptors, so they can actually intercede in this electron transport chain and put in some of these acceptors that are made in the laboratory, and they can actually accept and take the electrons away from the electron transport chain if you put in enough of these artificial acceptors. Okay, so does everyone understand about these electron transport inhibitors? They block the electron transport chain, therefore you cannot obviously reduce oxygen, and also you cannot create a proton motive force. Okay, the next class that I talked about were the ATP synthase inhibition. Okay, and I'll just go ahead and give you a couple examples of those. One of those ATP synthase inhibitors. Okay. One is called oligomycin. And that inhibits the F1 portion. Okay. And another is called dicyclocarbodiamide. Dicyclocarbodiamide. diimide. And this is sometimes abbreviated as DCCD. What does this do? Well, it looks like this. Let's see if I can draw this correctly. Bless you. And this reacts very well with carboxylic acids. And so you could have a carboxylic acid that comes along, and it will attack it will attack right here, okay? And there's several other reactions after this, which I don't have to give you right now, but it actually attacks these carboxylic acids. And remember, this is a very good inhibitor for the F0 part of the ATP synthase. So can anyone guess as to what amino acid would be especially susceptible to DCCD inhibition? Aspartate. Very good, aspartate. Can anyone name another one? Glutamate. Very good, very good. And do you remember I talked about how these protons get transferred in the F0 part, right? It gets transferred from one carboxylic acid to another. Now if you block these carboxylic acids by this DCCD, it can no longer carry those protons through the F0 part, and therefore you cannot rotate that gamma subunit that's essential for ATP synthesis. Okay? So this will block rotation of the gamma subunit. Okay, so these are ATP synthase inhibitors. There's another inhibitor called uncouplers. Okay. And uncouplers are kind of sophisticated, okay? So what uncouplers do is that they dissipate the proton motive force before it has a chance to create ATP. Okay? I'm going to use that word dissipate. Dissipate the proton motive force. Okay. How do they do that? I'm going to draw for you the matrix. Okay. This is the inner mitochondrial membrane. This is the intermembrane space up here. And so what these uncouplers do is that they'll actually take the protons that are building up in the intermembrane space, and they will transport them back into the matrix. Okay? So as soon as those protons get translocated by the electron transport chain, they immediately get taken back into the matrix. And so now you no longer have the proton motive force, and you can no longer create ATP. One of the classic uncouplers is dinitrophenol. Dinitrophenol. I'll go ahead and write that out. Actually, it's called 2,4-dinitrophenol. Okay. Looks like this. Does anybody remember from your organic chemistry, these NO2s, are those electron donating groups or electron withdrawing groups? Beautiful. They're electron withdrawing groups. And what they do is that they lower the pKa of the alcohol here so that proton can easily leave. Without these nitro groups here, without those, then the pKa of the hydroxyl group is about 10. So if you look at tyrosine, which has very similar side chain there, the pKa of the proton on the alcohol group is about 10. But now that you have these electron withdrawing groups, it actually lowers the pKa, so this proton comes off pretty easily, okay, at about pH 5, pH 6, okay? And so now you create this. Okay? And that's a negative sign up there. Okay? Well, it turns out that the pKa of this 2,4-dinitrophenol is perfect for capturing the proton here in the electron in the intermembrane space, and then once it drags it across the matrix has a high enough pH so that that proton comes off. And then the 2,4-dinitrophenol can go back and pick up another proton and bring it back across the matrix. Okay? And it turns out that in the 1940s, people started to notice that you could use this as a diet drug. Okay? And it was used to lose weight. Lose weight, how could that happen? Well, let's think about it. 2,4-dinitrophenol, what it's doing is that as soon as the NADH comes in and releases its electrons, okay, it's going to dissipate that proton motive force, so no ATP can be created. So what does that do? You start to build up ADP, okay? And ADP is going to do what to the catabolic pathways? Is it going to activate the catabolic pathways, or is it going to inhibit the catabolic pathways? ADP. Is it going to inhibit or activate? It's going to activate, because that means low energy charge, right? Low energy charge, you're going to activate. So then your body's going to want to break down glycogen. And then after that, it's going to want to break down, what's the next thing? Fat, right? It's going to start to break down fat. And so all this glycolysis and Krebs cycle and the fat oxidation, all that's going to do is create more NADH. And that more NADH is going to feed into the electron transport system, okay, and it's going to get oxidized. It's going to make protons go across the membrane there. But as soon as those protons go across, boom, the proton motive force is dissipated by this magic molecule, 2,4-dinitrophenol, okay, and you don't create any ATP. So again, your ADP levels increase, your ATP levels decrease, and you start to break down more and more fat. Seems perfect, right? Gosh, I want to have it. No, but the bad part, there's some bad part here. One of the bad things that happens is that these poor individuals that took these pills, they started to have to pant all the time. Panting, means you're basically inhaling, exhaling a lot, okay? Why is that? Why are you inhaling, exhaling a lot? Excuse me? Because you need oxygen, right? You keep on consuming oxygen because the electron transport chain is going like gangbusters, right? It just keeps on. You have tons of NADH that's being created. It's going. The electrons are going through the electron transport chain. You're breathing in that oxygen to reduce it to H2O, because that's the final electron acceptor, okay? And so these poor individuals would go around constantly heavily breathing all the time. The other thing that happened to these people is that they would get fevers. And it turns out that for some reason, I don't know exactly why, but transporting these protons across this membrane in the opposite direction increases the temperature of the body. And as a matter of fact, bears in hibernation, they actually have something similar to 2,4-dinitrophenol in order to keep their body temperatures high during the cold winters. Okay? They actually use a protein. They don't actually use a small molecule called 2,4-dinitrophenol, but they actually keep themselves warm by causing these protons to come back across the membrane. So all these bad side effects, and of course some of these individuals died because they overdosed on these 2,4-dinitrophenol pills, and so they took it off the market. And there's been similar diet pills even after 2,4-dinitrophenol that have operated on the same principle, because you really do lose weight, okay? But the side effects are too severe. Okay, so I've talked about the uncouplers. Oh, there's one more, and that's called ATP. ATP, ADP exchange inhibitors. ATP, ADP exchange inhibitors. Okay, and one of these is called atractiloside. Let's go ahead and write this out for you. Atractiloside. Okay, and you might be thinking, well, how could that cause inhibition of this whole thing? Well, remember, when we're inside the matrix here, okay, you're building up ATP on the inside, okay? So ATP is being built up on the inside. And you're consuming ADP, okay? And so at some point, you need to get that ATP out for the rest of the cell, okay? You need to get that ATP out. And you need to bring in ADP to be used for more ATP synthesis, right? So you need to bring in fresh supplies of ADP. You need to remove the ADP. Well, there's a protein in there that exchanges those, okay? And so that exchanger, if you inhibit that exchanger, then ATP starts to build up, and you have something called product inhibition. Product inhibition. And so the ATP synthase can no longer release that ATP, okay? And the whole thing slows down in terms of ATP production. So you got to be able to get that ATP out. If it builds up to too high of a concentration, it's going to shut down ATP synthesis. Okay, so we've talked about this system, this beautiful system. I think I have time to go over this handout with you. So if you have not gotten the handout, please pick one up from here. We'll go through this. Okay, so what I did here is I went ahead and sort of summarized the last few weeks of catabolism for you. Okay? And in catabolism, the whole goal, of course, is to make ATP. And I thought we would go through this very carefully. So what I did is I separated this into different columns. We have metabolic stage, NADH, that's produced at that metabolic stage, the enzymatic reactions that are involved, FADH2 if it's been created, the enzymatic reactions involved in FADH2, and then ATP if it's been created, and the enzymatic reactions involved in that, okay? So let's just talk about glycolysis. So glycolysis is the first thing we talked about. And as I said, and we've talked about, we make 2 NADHs, and those are going to be in the cytoplasm because that's where glycolysis takes place, in the cytoplasm. And the enzymatic reaction involved is glyceraldehyde 3-phosphate going to 1,3-bisphosphoglycerate. Okay. Is there any FADH2 created in glycolysis? No way. You guys should all be shaking your head no. Okay? Good. Okay, and so there's no enzymatic reactions involved in that. But we do know that there is ATP created from glycolysis. There's 2 of them. They're found in the cytoplasm. And the enzymatic reactions that eventually make the 2 net ATP molecules, okay, the 2 that we finally get out, is going from phosphoenolpyruvate to pyruvate. Okay, so that all makes sense. Then we have this enzyme called pyruvate dehydrogenase complex. It's that massive enzyme that has 3 subunits, and it's got 5 cofactors. Okay, it's found in the mitochondria. Okay, we make 2 NADHs from that. We don't make any FADH2s. We don't make any ATPs. Then we come to the citric acid cycle. Citric acid cycle, we make 6 NADHs. Now we're counting glucose as our starting material, right? So from glucose, let's review this. How many acetyl-CoAs do we make from 1 glucose molecule? 2, right? So 2. So I remember every 1 round of the Krebs cycle, you get 3 NADHs, but because we're starting off with 2 acetyl-CoAs, we will make 6 NADHs total. Okay, so 6 NADHs. These are the 3 reactions that are going to make those. Isocitrate going to alpha-ketoglutarate going to succinyl-CoA, and malate going to oxaloacetate. We also make 2 FADH2s, okay, and that's also made in the mitochondria, and it's going from succinate to fumarate. Okay, and of course we also make 2 GTPs, okay, that's made in the mitochondria. That's going from succinyl-CoA to succinate. Okay. By the way, we specified this type of phosphorylation here as what? What level of phosphorylation do we call this? Substrate level phosphorylation. Very good. Okay, so we've got our subtotals here. I've got 2 cytoplasmic ATPs made, 2 cytoplasmic NADHs made, 8 mitochondrial NADHs made, 2 FADs made in the mitochondria, and then 2 cytoplasmic and 2 mitochondrial ATPs. I'm doing a separation here because it gets a little complicated when we start talking about different tissues. So bear with me about separating the cytoplasmic versus the mitochondrial molecules. Okay, now let's go ahead and talk about oxidative phosphorylation. So we're going to be talking about specifically the heart and the liver. So in the heart and the liver, we're going to create 2.5. We have to multiply 2.5 for every cytoplasmic NADH. So we get 5 ATPs for that. 2.5 for every 8 mitochondrial NADH. We get 20 ATPs for that. And 1.5, we don't get as much. We don't get as much ATP out of FADH2 because you guys know that the standard reduction potential for FADH2 is not so negative as it is for NADH, right? So you can't get as much energy out of FADH2 from the standard reduction potential. Okay, so we only get 1.5 for every FADH, okay? Every FADH2. So we get 2 FADH2s, that's going to be 3 ATPs. And then here I wrote substrate level. These are the ATPs made by substrate level phosphorylation. So I get a total of 32 ATPs here. Okay, and I have this caveat. So this caveat assumes that the NADH gets transported into the matrix through this particular pathway, OAA going to malate, and then the malate gets into the mitochondria. And that is actually true for the heart and liver. There are other organs that are not so great at getting the NADH that's created in the cytoplasm into the mitochondrial matrix. For example, the skeletal muscle in the brain, for some reason, Maybe it's just an evolutionary glitch. I don't know. I always get the question, why is it? Why is it the brain is so inefficient? I don't know. But for some reason, these do not have good transport systems for getting the NADH from the cytoplasm into the brain. From the cytoplasm into the mitochondrial matrix. So what happens? What happens is that the NADH actually can only create 1.5 ATP molecules for those cytoplasmic NADHs. Remember I had talked about coenzyme Q, and I showed all those arrows going to coenzyme Q in your book, okay? So it turns out that in the skeletal muscle in the brain, there is a system for NADH to go to an enzyme that's on the intermitochondrial membrane, and that enzyme converts it to FADH2, and then FADH2 donates those electrons to coenzyme Q, and that's what's happening in the skeletal muscle in the brain. So we don't get as much out of the NADH that's created in the cytoplasm. Of course, though, those NADHs that are made in the mitochondria, okay, are good. Okay, so they're going to make their full 2.5 ATPs for every mitochondrial NADH. So we have 20 ATPs there. Everything else is the same as it was up here. So in those particular tissues, you're going to get 30 ATPs compared to heart and liver, where you get 32 ATPs. Okay. Any questions about this? All makes sense, but I don't want you just to memorize it. I mean, just know it, but I want you to kind of understand the logic. That's really important. So understand the logic, why it is that we're getting these ATP molecules, and of course, this factor comes, this 2.5 and 1.5 comes from where in the complex, where in the electron transport chain the electrons are feeding into. Okay, we will stop right here. We will have a midterm on Monday. Please get here on time. Bring a ruler, bring a calculator, and study the chapters since the last midterm. Okay, so we had left off, I think, in the middle of 12. I don't think we actually got all the way through 12. So I had to start off like somewhere midway in 12. So 12, 13, 14, and 15 will be up for midterm number 2. Remember, if you guys improve, I will not emphasize so much the first midterm. So you guys have a chance to do well in the course still. Okay. Have a good but studious weekend.